Epilepsies. This is my of note. This is my first presentation of regarding the genetics of neurodevelopmental disorders. I'm sure that in the audience there is much more expertise, and of course, feel free to interrupt me if you have something to add or questions. So, for introduction, quickly define epilepsy as a recurrent which comprise a clinically complex group of neurological disorders defined by recurrent spontaneous seizures alongside abnormal excessive neuronal activity in the brain. It has a global prevalence estimated in the range of nearly 1% of the entire population. And it has been estimated that 1 out of 26 individuals will develop epilepsy at what point during their lifetime. Well, there is a broad spectrum of phenotypes and comorbidities regarding epilepsies. We distinguish three main subtypes that we are going to investigate here. First, the genetic generalized epilepsy, which is featured by generalized seizures all over the brain, and alongside adult focal epilepsies, AFI, and Rolandic epilepsies, which are featured also by focal seizures, not in the entire brain. Within this type of epilepsies, a strong genetic component is recognized. Recent estimations have suggested an undisputed proportion of this genetic component that is at least 95%. Within the non-genetic component, we know that it's highly heterogeneous, having common variants with low to modest effect size and two highly effect size variants with very rare frequencies. Among this entire spectrum, a lot of genes has been associated, mainly to singlet nucleotide variants, there is SNVs, but also copy number variants has been associated. The latter are defined as gains or losses of genetic material with comparison to the reference genome. Given the size of these variants, they are usually located here, the genetic associations, and are usually rare and de novo, with a de novo behavior. A lot of reports mention and have associated CNVs to several types of epilepsies, but they are usually concentrated in specific locus upon the genome and with highly comorbid with other neurodevelopmental disorders, particularly with intellectual disability, schizophrenia, and autism spectrum disorders. Well, the objective of the present study is to first assess the role of rare and large microdeletions within and outside of hotspots, which is given by this blue ring here. And then to study the risk conferred by CNVs in common epilepsy subtypes described before, such as GGE, AFI, and Rolandic epilepsy. We assembled a large cohort of epilepsy patients and controls, which reached a total amount of 9,200 samples, including 2,454 cases of epilepsies. These samples come from two different platforms, Affymetrix Generalized 6.0 and Illumina Omnix for the focal epilepsies. These two last ones are not reported yet. The criteria for inclusion includes first that the copy number variant must be a deletion type or with a deletion behavior, that is microdeletions. They must be rare with a frequency below 1% and large, above 400 kilobases, and also must include at least 100 proofs involved in the genetic variant correlate. Overall, a total number of 345 microdeletions were detected and further analyzed. Basically, we analyze this variance comparing the presence of microdeletions among patients versus the presence of these microdeletions in controls. And we first assess a combined epilepsies analysis using a generalized linear modeling, studying first all microdeletions, counting each patient as one. That is 234 of the 2,000 cases presented at least one microdeletions, in comparison to 133 patients with at least one microdeletions in controls. At the beginning, we see a strong enrichment of all microdeletions. And then when we assess specific subsets of microdeletions, like CMBs overlapping constrained genes that do not tolerate gains or losses, according to references databases like the database of genomic variants, which count this CMB set, and also overlapping neurodevelopmental genes, autism spectrum disorder related genes, and this is wrong, but this is developmental disorder genes reported in the DD2G database, and loss of function intolerant genes coming from the recently published exome aggregation consortium. Finally, also CMBs overlapping hotspots loci, and as a control, a set of random genes reported previously. We see that the strongest association comes from the CMBs overlapping hotspots loci, and following immediately by neurodevelopmental genes. This is in the combined datasets. Considering these results, we decided to evaluate subtype-specific burden analysis, considering only the subtype cases for each dataset. As we can see, here we have the odds ratio, which denotes the effect size or the actual risk that the presence of at least one copy number variant within these cases confers to the development of the disease. We have the same CMB sets here, overlapping neurodevelopmental genes, autism spectrum disorder related genes, developmental disorder genes, and loss of function intolerant genes, coming from EXACT again. We see that only GGE reach significance in all microdeletions subset analyzed. That is, when we have an odds ratio of 1, we don't have an actual risk of the genetic variant or the pool of genetic variant that we are assessing here. And we again see that the strongest signal comes from neurodevelopmental genes and also from hotspots. Rolandic epilepsy exhibits a trend, but not significant, from autism spectrum disorders related genes, and no signal is observed for adult focal epilepsies in neither of all subsets. Then, given our results, we decided to analyze the burden that we could actually observe outside hotspots. And we can see that the signal is quite strong here, with an odds ratio above 7 for genetic generalized epilepsies, but not from any other disease assessed. Only the subset of copy number variants in neurodevelopmental genes survives association outside hotspots. And then, given our results, we tried to ask if there is any gene enriched with microdeletions outside hotspots. We took first, we assessed first nominal association, comparing the amount of genes touched by CMBs in cases in comparison to controls. And we first got nominal association for RB-FOX1 and Norexin-1, which of course will never survive genome-wide significance, but still neither of them are observing controls. And using the same criteria, we managed to extract a set of 12 candidate genes that are recurrently more than once deleted in cases and never observed in controls. It is important to mention that some of these genes have been associated with SNVs, for example, protogathering 7, to epilepsy, and we observe that the presence of these genes are also present in different types of epilepsies. Well, in sum, we conclude that microdeletions contribution to epilepsy outside hotspot loci might be overestimated, and their contribution for large microdeletion might be lower than expected outside hotspot. And also, we see a clear trend for microdeletions giving risk only to genetic generalized epilepsies, followed with a minor contribution of Rolandic epilepsies and never seen in adult focal epilepsies. Still, recurrent genes are present outside hotspots and also commonly observed more than once in different types of epilepsies. So, in this regard, this is part of an ongoing study that is currently on revision, and there is a lot of partners, and of course this is a collaborative effort of many authors and institutions, which I'm very grateful to, and particularly to Dennis to introduce me to this epileptic genetic world.
Eduardo, can you hear me? Is it acceptable to interrupt with questions, Dennis?
Of course.
Can you go back one slide, Eduardo?
Yes. Which slide?
Yeah, that one. Perfect. Yeah, I just wanted to point out something interesting you probably already know, but I think it's an area of research that's underappreciated. The gene on your slide called T-SNARE1 is a GWAS hit in schizophrenia, and it is not a usual snare protein. No one knows what this protein does, and I think it's part of the Golgi. I can't remember that part for sure, but nobody has really worked on it in schizophrenia, to my knowledge, to try to figure out what its function is. But I know for certain it's not a usual snare protein, which makes it interesting, I think. So I just wanted to make you aware of that.
Yeah. In this regard, it is important to mention that almost every gene associated to epilepsy, whether by a single nucleotide variant or copy number variant, are also, it's commonly observed with other traits highly comorbid with epilepsy, like intellectual disability, autism spectrum disorders, and also schizophrenia.
Yep.
So it is not strange to see it there.
Okay, thank you. Eduardo, if you want, you can stop sharing the screen. So,